In this video, we will discuss about second generation CAR T cells. So that can be used for our future therapies to treat the patients that are affected with cancer. So in one of our previous videos, I have discussed about CAR T cells, first generation CAR T cells. So in this video, I will discuss in brief about second generation CAR T cells. So what is the difference between first generation and second generation CAR T cell is this linker. In the I will not discuss I will not discuss about the first generation CAR T cell linker, but I will discuss here only about the second generation CAR T cells. So this is the antibody that has been uh, produced against the specific antigen, and the way that these antibodies can be produced is this monoclonal antibodies uh, produced is by using immunogenic some kind of immunogenic animals where they immune those animals against the antigen of our interest, and the antibodies are generated against in those animals, and then from there they can purify those antibodies and link them to this chimeric antigen receptor. This is the whole receptor in the form of chimeric antigen receptor, and then this uh, chimeric antigen receptor can be targeted. This chimeric antigen receptor can be used to target against those antigen within the host. So what happens when this CAR T cells is expressed on the cell surface like it is shown here is that the top part is the antigen top part is the we can say single chain fragment, or in other terms, if we go in detail, it contains variable heavy chain and variable light chain that is linked together with the help of this linker. So what it does is whenever it has the ability to detect the antigen, specific antigen, as it is generated against those specific antigen that are present on the tumor cells. So whenever it detects the antigens that are present on the tumor cells, the intracellular domain part, there are two intracellular domain part. First one is co-stimulatory domain, another one is CD3 domain. So both of them activate both of them has different form of activation. The CD3 domains activate whenever whenever the CD3 domain activates when the phosphorylation of the CD3 domain occurs, and it forms a complex with its ZAP70. What it will does is it will trigger this transcription of T cells effector function, or in other terms we can say IL-2 proliferation cytotoxicity. So it will triggers this complex ZAP70 and CD3 phosphorylated complex will trigger this signaling cascade that will leads to the transcription of T cell effector function, or the production of IL-2 proliferation and cytotoxicity, and that in turn will leads to this will leads to the release of this perforin and granzyme in the extracellular matrix. And what it does is this perforin can enters into the tumor cells with the help of pore, and then it will cause the cell death. So this is how the second generation CAR T cell works. And this is just, and here on the top left side we can see the how this single chain fragment uh, formed. So the antibodies that are generated within the uh, animals, the part of that antibody, that is variable heavy chain and light chain, is been used to form this chimeric antigen receptors. And this co-stimulatory domain activates via different signaling pathway. However, whenever this co-stimulatory domain activate, it will also finally leads to the transcription of T cells effector function, that is IL-2 proliferation cytotoxicity, and that in turn leads to release of this perforin and granzyme, and that will enter the tumor cell and cause the death of the tumor cells. So this is a second generation CAR T cells idea that can be used to treat the patients that are affected with tumors, especially the solid tumors. So that's all for this video. If you need to understand more about this second generation CAR T cell, you can go through the review paper that has been provided in the link has been provided in the description. That's all for this video. Thank you for your time.